So the second speaker for our session today is Ben Cole. So Ben is a research scientist at the U.S. Department of Energy's Joint Genome Institute in the Lawrence Berkeley National Lab. So he is my go-to person often to talk to about metadata. So he's going to talk to us today about the best way to handle and report single-cell RNA-seq or single-nucleus RNA-seq metadata. Thank you so much, Ben.
Thanks, Rachel. And it's a pleasure to be here and present. And I also want to thank Fabio and Kevin for inviting me to talk today about metadata. And this will expand a little bit on some points that Bert already introduced in the first talk today. But I also wanted to give a little bit more practical advice for how do you generate, how do you handle metadata that's coming out of your project, and also try to motivate everyone to really think about metadata more in their single cell research. To preface my talk, I wanted to say that in plants, single cell research has been lagging behind a little bit animal single cell research. However, over the past couple of years, I believe that we've reached kind of an inflection point where we're generating now, millions of cells per year from a variety of different species, and it's only going to go up and up. And so I did a survey of the plant single cell literature recently last year, and found over 50 studies that have generated single cell data, a lot more now. And this encompasses 6 different single cell sequencing platforms, over 350 samples, a variety of tissues and species, and as well as treatments. So I think that it's really important to get a standard for how we report our data and metadata now, rather than wait another few years when we're so saturated with poorly annotated data that it's going to be impossible to kind of catch everything up to a future standard. And so all of this recent research has resulted in now resulted in some reference data sets, which I'm really excited about. And these are 3 of my favorites at this point. The first is one that I had the pleasure of participating in from Philip Benfey's group, and Rachel and Chawei were leading the generation of this root atlas in Arabidopsis. It's really foundational in a lot of ways, including the really detailed way that these trajectories and cell types were specified. More recently, Ken Birnbaum's group has generated a grass atlas that encompasses 3 different grass species, which are historically a little bit more challenging to work with, but they did a really fantastic job in annotating root cell types in a pretty clever way across all of these species. And then recently, this tour de force from Joe Ecker's group that followed Arabidopsis development from seed to seed, generating this absolutely massive data set of over 800,000 pre-processed cells and 430 so-called high-quality cells that will really be a really nice resource for the plant community moving forward. Given these reference data sets, what we want to do is make our single cell data useful to the rest of the plant biology community. You've spent a lot of time, a lot of money, a lot of consternation and anxiety over getting your protocols optimized, getting your analysis workflows done, that you want to make sure that your data is useful and, of course, more highly cited, for many years to come. But more imminently, I wanted to present this sort of motivating scenario for why you should think about metadata. In this scenario, you've just hit the submit button to this really landmark Nature paper that finally figures out what auxin does in plants. And this data set, features millions of cells, has spatial validation and mutant analysis. It's a real, a really nice article that's going to be very high impact. And of course, you posted it on bioRxiv because you're really excited about it and you wanted everybody to read it before it goes public. And then to celebrate, because you've just done like 3 years of hard work in the lab and at the computer, you take a backpacking trip to Iceland to try to relax and get some R&R while the reviews are tearing your paper to shreds. But unfortunately, while you're on backpacking trip, you lose all your data. Either somebody steals your laptop, or you drop it into a volcano, or something happens, and all this hard work that you've done is now literally up in flames. So you're bummed, and you come back, and you're kind of bolstered by the good reviews that you got. But one of the reviewers wants you to generate like 50 new plots of gene expression profiles from your data, and you don't. And the question is, do you have enough of your analysis pipeline? Is it reproducible enough that you can go back and prepare those to finally get your paper submitted? So really, how prepared are you to regenerate and reproduce your results that you've put out there? And so I want to take a step back and describe like what do I mean by data and metadata? So data to me represents the primary information that's generated by lab instrumentation. In the single cell world, this is FASTQ files and other data that sort of goes along with that. And also it includes information directly processed from the primary data into tables, matrices, etc. So this is can be hundreds of gigabytes. It's a really unwieldy data set, but it's the foundation of your work, right? Of a single cell sequencing study. What I mean by metadata is all the information that describes the data. And this can go from how the data was generated, quality control metrics, insights that are gained from analysis of the data. And this is usually, or ideally, structured in a way that everybody will understand. So it has common terms describing like what that metadata actually is. So for example, like a cell type would be a structured term for describing an inference that you've made for a particular cell. And in the ideal world, both data and metadata will be both human and machine readable using a standard architecture. And I'm sorry to say, this is actually pretty rare in the field now. Rare in that a complete set of metadata is reported in a repository that's machine readable. And I think that there's a lot of room for improvement in the plant, especially in the plant community for this. So so metadata follows every single step in the single cell workflow. It starts at sample collection. So you need metadata that describes how your plants were grown, what conditions you used to collect your initial tissues, what experimental factors you used. Did you stress them? Did you grow them on soil? Did you or different growth substrates? How they relate to other samples. Were they batch grown with other samples that were processed at the same time? What particle are you isolating? Bert did a really nice description of some of the pros and cons of going from cells or nuclei for single cell analysis. But this is a really important parameter that people will need to know because it affects how you think about the data. And then what method did you use to prepare your particles? And did you do any QC on those? Did you do a viability test? Did you do any other assay that tells you how high quality your sample is? So all these describe the sample, so the collection of cells that you're subjecting to single cell sequencing. Other metadata describe the library that the method that you use to take that cell population from that sample and process it into a single cell data set. And so this is critical information like the kit type that you use to generate your libraries, the version, the index. This is a critical point for especially for 10x. There's 2 different versions that are floating around that have different read structures that influence how you can reanalyze your data. And then how, was there any library QC that's done? I find that very rarely do people actually report, for example, bioanalyzer traces of their libraries in any kind of public repository. This could be really useful for somebody who's just starting out in doing single cell analysis and want to look at what a typical or high quality trace will actually look like to decide whether they actually want to sequence their sample or not. And of course, what the read structure of the sequencing library is. And then the actual sequencing data. So this is probably the most important component of your data set. So this is the raw sequencing data, usually in a FASTQ format. But there are other formats that can also be used to reconstruct this raw sequencing data. And then all the metadata that's associated with sequencing, including the platform used to sequence it on, read counts, the quality of the read, accession numbers if they're public, deposited in a public repository, and then which repository they are in. So I find that for most single cell studies, this is usually pretty good. People usually put their data into a repository with metadata that's associated with the actual sequencing, because a lot of funding agencies, journals require this information. There's this enforcement mechanism that exists already. I think the most lacking form of metadata out there is analysis-related metadata and data. So the data itself, the barcode, gene barcode matrix that you process your FASTQ files into, and this will be introduced and explained by later talks today. So this is usually deposited, but there are processed or filtered cell data objects that are more rarely deposited in these repositories. And then the metadata associated with these matrices that include everything that you've inferred from your cell. This is probably one of the most exciting aspects of your analysis. All that hard work that you did to analyze cluster by cluster, with validation. It's really important to make this data available so that others can use it, use your data to help describe their work. Cell level metadata includes cell type and associated metrics. If you've done a pseudotime trajectory inference, that's a really important thing to report, as well as the metrics associated with that. And by metrics, I mean, how confident are you in those assignments of either cell type or pseudotime? And then other cell features like, if the cell is dividing or not, as Bert mentioned, this is something that can influence how a cell is being clustered. And then most important for the cell level data is to include the actual barcode, the sequence barcode that refers to that particular cell, so that someone could take that metadata and then reconstruct your analysis pipeline. Also important is all of the code that you use to generate your analysis. This is, Git and GitHub are a really good resource for publishing your code, and also having a version control over your code. So as you make changes, it will all be tracked and documented. And then also, lastly, the analysis environment that you use to run your code. Some pieces of software run differently depending on the architecture that they're run on. So it's important, it's helpful to have your analysis environment very well documented at the least. And I'd say one of the better things to do is use a container system like Docker, and I'll explain that in just a little bit. So an important point is, across all of these metadata and data tables, it's important to have common identifiers that link all of the steps in your workflow. So in your cell level metadata table, I should be able to know what sample that cell came from, how it was processed, and that will allow me to go to other metadata tables to figure out how it was processed and sequenced, et cetera. And then I can't stress this enough, but you can never have enough documentation for yourself and also for others, to really describe what you did. And where possible, try to make things machine readable, at least, and also available via fixed URL or through an established API. And avoid relying on supplemental tables and articles, which can be paywalled, as well as non-standard data format. So some of my favorite tools, Rachel, am I running out of time?
Yes, I'm sorry to interrupt you, Ben. Maybe just like 30 seconds or one more minute,
Okay.
so we have time for questions.
Okay, so I just wanted to introduce some of my best friends for handling metadata. These include the Gene Expression Omnibus, NCBI. If you're using R, Targets is a really nice way to document your workflow. Docker is a really nice tool for documenting your analysis environment, and of course GitHub for your code. And then really quickly, I just wanted to go through two recently published data sets that I think did a really nice job in documenting their metadata. The first is this study by Trevor Nolan from Philip Benfey's group that did a really nice survey of rhabdomyosteroid and Arabidopsis roots. So, good, data is available in a public repository. The data is also well described, including metrics with R objects, and all of this is available in a Shiny browser. And the metadata, but some things that could be improved is that metadata is contained in these really large R object files versus a lightweight CSV that could be a little bit more easy. And the sample information is available in a supplemental PDF, which can be hard to access. And the second example is this study from Bruno Guillotin from Ken Birnbaum's group. Also, the data is well documented in the public repository. There are metrics available, sample descriptions. Cell metadata is also included as a standalone CSV, which I really like. But some things that could be improved are, the sample description itself could be deposited in Geo, so it's more easily accessible, and the analysis code or environment could be made available as well. And so here are some other reasons for why it's important to meticulously document your metadata. The I and R in FAIR, interoperability and reproducibility. And I also wanted to say that there's a lot of progress on this from EBI as well as others to provide formats and standards for documenting your metadata. I'm sorry I went over time, but I can take a couple of questions.
Thanks so much, Ben. So we have a question. The first question in here, I think, is more of a technical question that maybe I'll follow up with a more general question that you can discuss this area. So someone is asking how to actually add a gene ID column in the metadata. So maybe you want to just talk about how might a wet bench scientist, for example, figure out how to add things properly to the metadata. And then maybe a shameless plug for the PCA. How might the PCA be providing resources for people to learn?
Yeah, I mean, a metadata table is really, well, so there's a variety of different formats that you can use, but the most common one is just a simple spreadsheet. So, you could make a metadata table in Excel. You could add as many columns as you want with, for example, if you want to associate gene IDs to inferred functions, whether it's a marker gene or not in your single cell data set, should be pretty easy to do in a spreadsheet format. There's, I'd say, not well enforced standards, but there are some, standards that are out there for how you can do that, using, different, annotations for the genomes, gene models for different genomes. So it could be as simple as that. And then for the PCA, I think PCA can provide resources and tools for, and also just general advice for how to format and deposit your metadata. But I also think that it could be have a role for advocating, more widespread reporting of this, particularly at the journal or funding agency level. We've got a lot of people in the PCA, which makes our voice kind of loud. And so if we really use that, I think we can maybe generate more traction to get things more up to standard.
Thank you so much, Ben. I think that's all the time that we have.